So as I said, today we have a lot to cover. We've got a good amount of notes to take, so I want you all to be in a good position starting at the beginning of the lesson, which is right now, to be able to capture the most relevant information. That way you can refer back to it as you do your asynchronous assignment and your exit ticket, and also as you begin to prepare for the unit exam. So we're starting our new unit, Unit 3. This unit is all about cellular reproduction. And this unit is going to go through next Wednesday, which means that you have until next Wednesday to complete all of your assignments from this unit. And you also should know that your unit exam will be next Wednesday as well. Just want to put that on your radar. Today's first lesson will be entitled The Cell Cycle Review. Today is February 2nd, 2021, and it is the first day of our third unit. The objective of this lesson is to analyze how cells grow and reproduce in terms of the cell cycle, which is made up of 3 parts: interphase, M phase, and cytokinesis. Thanks for joining us, Sienna. Two questions that we seek to answer by the end of today's lesson: why do cells reproduce, and how do cells reproduce? This second question, how do cells reproduce, is one that we will seek to answer over the course of this entire unit. So keep that question in mind. Why cells reproduce? We're going to talk about 3 basic reasons why cells reproduce today. So make sure you've got some information from this slide and you're ready to go. All right. First of all, I want to celebrate, wrong class. Give me 1 second. Got to do this. All right, first of all, I want to celebrate our classmates who mastered the exam yesterday. We had quite a few people do it. 9 of our classmates were able to get an 80% or above, and that's even before the curve. So we did really, really well yesterday. I want to congratulate you all. That's really a remarkable achievement this early on in the course. We've got to keep this momentum and build off of it. The people whose names are on the screen did really well, but there's still room for improvement. And those of you whose name is not on the screen, whose names are not on the screen, you all have room for improvement too, but I don't want you to feel discouraged. If these first 2 exams were really difficult for you, that means that you still have some restructuring to do. That means you might want to think about the ways that you're studying, think about the ways that you're note-taking, and think if you can do anything to edit those methods in order to improve them. So these people, Sienna, Janelle, Carolyn, Abigail, JT, Ashley, Josue, Dejan, and Melissa, excellent job. Definitely proud of you guys. Keep going. And it's, like I said, it's remarkable that we've got 9 people in the second unit. By the time we get to the end of the course, I have no doubts that everyone in this class has the potential to be at a mastery level. And the goal is for us all to master the EOC. We're going to get there. It's just going to take time, diligence, making sure we're doing our warm-ups, exit tickets, and asynchronous assignments, making sure we're taking notes, if necessary, making sure that we're consulting with Mr. Rudd to get some additional help. But good job. We're off to a great start. Yes, Lance, you did just say this in the chat, but Lance and Sully, you two, Sully's not here right now. Lance, you've got to take this test. We can do it after school if you're available today, but here's what I'll do. I will send you the link where you can schedule an appointment with me, and we'll make sure that we take the test in that time. All right. I will be updating grades this evening, so you'll see some movement in Power School. You should have seen some movement yesterday as I put in the Unit 2 assignments. I just didn't score them yet. So I'm going to go back in and I'm going to put in your grades for those Unit 2 assignments today. If you know you've got some missing assignments, then you can expect that some of those will be marked 0. Or if you know you did them, you need to go back and do them today. You obviously can still do that, but They will be marked late. All right. And then I wanted to just give you guys the opportunity to see some of our summary data so far. So when we compare Unit 1 to Unit 2, let's focus at the bottom here so we can look at our class specifically. Third block. In Unit 1, sorry, I'm waiting. There it is. In Unit 1, our test average was 70.27. We had 20 test takers. So far in Unit 2, like I said, we're missing 2 test takers, but our average is higher. It's 74.91. That's excellent. Our average went up. And I want to continue to see this improvement until our average is, as a class, above an 80%. I think we can definitely get there. We can also take a look at the number of students who got A's and B's on the exam. That improved from 9 students in Unit 1 to 13 students in Unit 2. So that's a good job. What you can see is that we not only took 2 of these students who had a C and boosted them up into the B range, but we also added a couple students into this A, B, and C range as well. So 51% of students were grade level proficient in Unit 1. In this unit, we had 53% of students. 24% of students were college and career ready in Unit 1, and 40%, almost 41%, were college and career ready in Unit 2. So we love this trend. We have to see it grow. We've got to improve from where it is. And I believe that we all have the opportunity to do that. All right. So our overall test average, when you take into account my other class, which is a standards class, was lower, but it's still higher than what it was. So I'm encouraging all of my students to keep pushing, keep going. We've got room for improvement. All right, jumping into the content now. So we're going to keep coming back to the Sterninger processes. This was a concept that we introduced in Unit 0, but it really is our guiding light for the entire course. Sterninger is so important. It allows us to capture the 8 essential life processes. In this unit, we're going to focus on one of the R's specifically. We're going to focus on reproduction. Reproduction. Reproduction is the production of offspring by a sexual or asexual process. Does anybody know what the difference between sexual and asexual is?
Isn't asexual kind of like making a copy of yourself in a way?
Okay, yeah. And Josue, were you going to add something?
Yeah, asexual is kind of like making a copy of yourself.
Yes, that's true. And that's what Lance said too. So we're on the same page there. Asexual is when one individual, one organism, basically makes a copy of itself. Sexual reproduction involves 2 different organisms. Typically, in some ways, it really just involves 2 different types of sex cells, which we'll talk about. But this process of reproduction is used to make new cells, and it's also used to copy and preserve DNA. Almost all cells, regardless of the organism in which they exist, will reproduce in their lifetime, including the cells that are in you. Cell division is another way of saying reproduction. Now, we've got some words that are highlighted in yellow here. Those are the words that I would like you all to write down. So cell division is the process by which individual cells reproduce. Lance, we can even do 3 o'clock today. I see you just scheduled your appointment. I'm meeting with another student at 3 who's also taking their test. So we can do 3 o'clock if you want to make it earlier.
That could work.
All right, and I will confirm that in email after this lecture. Okay. So here are some cool graphic images taken from a microscope that show cell division in action. On the left, we can see a plant cell dividing. Those big orange neon, those neon orange things, those are nuclei. And the orange things that are being split apart are chromosomes. We'll talk more about that. On the right, we can see a colony of bacteria growing in size and growing in population size very rapidly. So very quickly, because of this process of cell division, one bacterial cell can become two, two become four, four become eight, eight become 16. So we're not talking about a linear growth here. We're not talking about 1 to 2 to 3 to 4 to 5. We're talking about rapid exponential growth. And this is why the body has to have, the human body and all animals have to have an immune system that can protect them from this type of invasion. All right, so we can see very quickly how only a couple cells can become hundreds of thousands of cells. So that being said, I want you all to think about these questions at the bottom of the screen. Number one, why are new cells created? And number two, what's the process? What do you all already know about the process of creating new cells? Why do cells need to create new cells? What's that about?
I think they don't want to like keep the old ones. They still keep the old ones, but they still want to create new ones so that your body can produce new nutrients and more new stuff in your body.
Okay, that's a good reason, Lisa. Thank you. So sometimes we just need to create more of something. And that's kind of getting into one of the primary reasons that we'll talk about later in this lecture. Sometimes our bodies need to create more. They need more of something. So we need more cells to do that, new cells. Any other reasons we can think of? Growth, yes, thank you. Thank you, Abigail. So organisms grow from the time they're born until the time they reach physical maturity. Typically, that happens when they reach an age old enough to reproduce. So there's got to be a lot of growth that takes place in that time, and that requires new cells being made. So that being said, how are new cells made? Do you all know anything about the process?
Cell division.
Yeah, the process is called cell division. Do you know anything specifically about cell division? Okay, if not. Did you want to add something, Lance? I didn't mean to cut you off.
Nope.
Okay. All right. We'll talk about that. That's what we're going to get at with today's lesson and for the next 2 days. Quick flashback before we move on. This is a question from Unit 1. Which of the 4 macromolecules is DNA? This should be a quick answer.
Protein?
No! DNA
Nucleic
is not protein. What'd you say, Melissa?
Nucleic acid.
Yes, DNA is a nucleic acid. And of course, the NA in DNA stands for nucleic acid. Wonderful. All right, so we do have those 4 macromolecules. Lance gave us another one, proteins. What are the other 2? Because we've got proteins, nucleic acids, and then what are the other 2?
Carbs and lipids.
Carbs and lipids. Thanks, Ashley. And each of them is made up of their own monomer. Does anybody remember the monomers?
Monosaccharides, glycerol, and 3 fatty acids, amino acids, and nucleus. Is that what that's called? Nucleus.
Yes, nucleotides. Good. So we got those 4. The nucleotides obviously are the monomer for nucleic acids. Excellent. We got to keep coming back to this. This is a cumulative course. That Unit 1 information is not going away. So we've got to keep tying it in to the future units. So good job on that. All right, another quick TBT. We talked about DNA in the past, but we've got some other terms that are pretty much synonymous with DNA. They have some important differences that we'll discuss over the next few slides. But when we see these terms, our minds should start to make the association to DNA. One thing we can also notice is that several of the words begin with CHROM, chrome. That also should be a trigger to us. Remember, I want us to create these mental cues. Work smarter, not harder. Some of you do need to work a little bit harder, but most of you just need to work a little bit smarter. When you see that chrome beginning, that chrome prefix, you should be thinking, this has to do with DNA, chromatin, chromatid, chromosome, chromosomes, right? So we should be thinking about DNA when we see that prefix. There are some important differences, as I stated. So let's take a look at some of these differences. On the left, we can see the inside of a nucleus, almost as if it's been kind of dissected in half. And what's left is what looks like a bowl of spaghetti. The spaghetti inside of that bowl is called chromatin. Chromatin is really random, disorganized, tangled DNA. And again, the words and phrases and sentences that are highlighted, I would really encourage you all to write down. On the other hand, chromosomes are condensed and organized DNA. So if I had a shoelace and I just wrapped it around my index finger hundreds of thousands of times, really, really tightly, I'd cut off the circulation in my finger and it'd be really painful. But it would be similar to how a chromosome is organized. We've got long strands of DNA that get wrapped around tightly around what are called histones. And those are basically just protein complexes. So we wrap the DNA around that, and it allows us to organize the DNA in useful ways, rather than just having it lumped up in a bowl of spaghetti that's really tangled and disorganized. So on the left, we've got chromatin, which is random and disorganized DNA. On the right, we've got chromosomes, which are more organized and tightly wound DNA. Chromosomes can be broken down. Chromosomes look like the letter X, essentially. Each half of that letter X is called a chromatid. Chromatids that are matched up with one another are called sister chromatids. They are identical copies of DNA. At the end of each chromosome, either at the very top of the chromosome or at the bottom, we've got a region called a telomere or a telomere, which is there to protect the chromosome from deterioration. So there are your telomeres. They're highlighted in red there. And then in the middle of the chromosome, where the two sister chromatids are actually connected to one another, that's a region called the centromere. Centro. So centro means center and telo means end. Like a telephone that allows me to call someone from another end of the phone line. So I do think you should write down, I know it's not highlighted, but it is inside of a highlighted box. I do think you should write down these definitions: telomeres, sister chromatids, and centromeres. All right. So we know that we talked about the cells in the last unit. We talked about different cell types. Which type of cell has a nucleus, just as a quick review? Which cell types have nuclei?
You could carry.
Good, thank you, Lisa. Yeah, and I see we've got Abigail and Jean in the chat answering that question also. Eukaryotic cells have nuclei. And inside of that nucleus is where we find the genetic material. That's where we find the DNA. So we're zooming in. We start off with just a cell, just a eukaryotic cell. We zoom into the nucleus. That's where we would find the DNA. When we zoom in further and take a closer look at the DNA, we find that the DNA is organized into chromosomes. The genetic material is organized in chromosomes. And then if we zoom in even further, we find that the chromosomes are just tightly wound DNA in the shape of an X. We'll talk more about this tomorrow, but humans have 46 chromosomes that are organized in 23 pairs. Okay. So most of the cells in your body are constantly dividing, aka reproducing. Not all cells. We've got neurons that will live basically as long as we do, as long as everything is okay. But this is why older people start to have slower neural processing abilities. And in worst case scenarios, might develop a disease like dementia. But most of the cells in your body will be replaced in a relatively short time. And they're constantly reproducing, in order to do that. When we think about the cells inside of an animal or inside of a sexually reproducing organism, there are two types. There are gametes, which are sex cells. In the case of animals, we've got sperm cells and egg cells. And then everything else is referred to as a somatic cell. So gametes are your sperm and your egg. Somatic cells are everything else. Your nerve cells, your muscle cells, your digestive cells, your skeletal cells, everything else that is not a sperm or an egg is a somatic cell. So please make sure you're getting these different terms written down, gametes and somatic cells. We will refer to them as gametes. All right. Getting back to one of our essential questions, why are new cells made? We did get a couple reasons from some of our classmates. Essentially, those reasons were growth, but one of the most important reasons is replacement. So Lisa, you started to get at this a little bit, but old cells die, and this is a natural process. In fact, there's a name for it called apoptosis. It's a weird word. I can send it to the chat. We won't talk about it in this class, but if you're interested, you've heard of this, apoptosis?
No, I was gonna say that old cells die, and you gotta.
Yes, good. Yeah. So old cells die and they have to be replaced. This is a natural process called apoptosis. Cells really should die. And in fact, if they don't die, that can become a problem for the body. We'll talk about that later on. But we make new cells in order to replace those older cells. The second reason that we make new cells is for growth. And this is exactly who said it? Abigail said it and Lisa hinted at it. So most organisms are going to grow in size over time. Like I said, typically organisms grow until they reach an age or they reach a physical maturity at which they can reproduce. Growing requires new cells. So it's not that each cell in your body is getting bigger. It's that you're making more cells so that your body can get bigger. And then lastly, reproduction, both asexual and sexual. We need new cells for these processes. Sexual reproduction, like I said, involves a sperm cell and an egg cell. Typically, those come from those come from different individuals. But in the case of some very odd animal species, some species of jellyfish, they actually provide both, essentially, what is the sperm and the egg. Asexual reproduction is when one organism makes a copy of itself. It is genetically identical to the original parent cell. So 3 reasons, essentially, why new cells are made: replacement, growth, and reproduction. So we'll quickly watch some of this video. Tomorrow we'll discuss exactly the stages that the cells go through in order for them to divide like this. All right, you get the idea. Now, the cell cycle is actually the process that allows for that to happen. The cell cycle is the process cells go through to make new cells. It's got 3 phases: interphase, M phase, and then cytokinesis. We'll talk more in depth about these 3 phases over the next few slides. But we can see that interphase is made up of 3 parts: G1, S, and G2. M phase could be mitosis, but it could also be meiosis. We'll talk about both of those over the next couple of days. And then cytokinesis finishes the process. So please make sure that you write down the definition of the cell cycle. And it's called a cycle because once a cell goes through cytokinesis and we end up with 2 cells, now they are both kind of re-entering the original cell cycle. They're both put back into G1. All right, and here again they are. Here are the 3 major parts of the cell cycle: interphase, which can be broken down, and we'll talk about that on the next slide; M phase, which could be mitosis or meiosis; and cytokinesis. Okay. So interphase comes first, and it is the longest phase of the cell cycle. For most of its lifespan, a cell will be in interphase. That means that it's doing whatever its job is, but it's also growing, and it's preparing itself to divide. Interphase can be broken down into 3 steps. G1. G might stand for gap. It could also stand for growth. It just depends on how you want to remember it. But during G1, the cell is growing. During the second step is called S, or synthesis. The DNA is replicating. We're making a copy of the DNA. And then the third step is G2. Again, might stand for gap, might stand for growth. Just depends on your preference, really. The cell is growing, and it's also preparing to split up its cytoplasmic organelles. So some mitochondria go over there, some go over there. Some vacuoles go over there, some go over there. Some ribosomes go over there, some go over there. It's preparing itself to be divided. But as the second step, synthesis, is very, very important. The DNA must be replicated before the cell can divide. The reason for this is pretty obvious when we think about the numbers. So as I said, human beings have 46 chromosomes. In interphase, when the DNA is replicated, we make a copy of the DNA. 46 becomes 92. That way, when the cell is divided, those 92 chromosomes get split up into 2 new cells, and each of the new cells will have the correct number, 46 chromosomes. If the cell tried to divide before replicating its DNA, then each of the daughter cells would only have 23 chromosomes instead of the correct number, 46. Okay, so when we're preparing for mitosis, we need to replicate the DNA in order for the resulting daughter cells to have the correct number of chromosomes. And here's a quick, easy representation of DNA replication. We can see that the DNA is unwound. If you're interested in this process, we go into much greater detail in AP biology, but it's much like a zipper. At the bottom of this zipper, we can see that the DNA is still attached. 2 strands of DNA, each side of my jacket is attached. Above this zipper, the DNA has now been split apart. So the zipper is actually an enzyme in this case, an enzyme called helicase. Remember, enzymes end in ASE typically. So there's an enzyme called helicase that comes through and it unzips the DNA. And so it splits up the 2 strands, and now each of the strands can be copied. And we can see that process taking place here. We start unzipping in the upward direction. And each of the blue strands is copied to create another orange strand. And then the end result is 2 new exact copies of each other, 2 DNA strands that are exact copies of each other. After interphase comes M phase, and this is when the cell is actually being divided. This is when the chromosomes in the nucleus divide. There are 2 types of cell division. They both begin with M, and that's why we call this process M phase. Number 1 is mitosis, which we'll talk more about tomorrow. This is used for asexual reproduction to make copies of somatic cells. Then there's also meiosis, which is used for sexual reproduction to make copies of gametes, which again are sperm and egg cells. mitosis and meiosis. Sound similar, but they're quite different. So this is kind of what mitosis looks like. We start off with one cell. That one cell starts to split, and eventually it migrates far enough to become two distinct cells. But remember, before M phase can happen, we have to make a copy of the DNA. I'm going to continue to bring that up. I will be asking you all that in so many different ways over the next few days. So please make sure you're clear on that. Before M phase, we've got to go through interphase because we've got to copy our DNA. And then lastly, cytokinesis. Cytokinesis literally means cell movement. Cyto means cell, kinesis means movement. This happens when the cytoplasm and the cell membrane of the original cell, the parent cell, split, and they create two new daughter cells. Janelle, I forgot to send you this link to rejoin the Remind chat with your new number, or you can text at S2RUDBHBIO to 81010. Just sent it to the chat. I'm going to remove this other person. Okay. And then here we can see briefly this process of cytokinesis. We'll see it again in greater detail tomorrow when we talk more about mitosis. But essentially, the parent cell splits up its cytoplasmic components, all of the organelles, and it splits up its cell membrane, and it becomes two distinct cells. Oops, I did not mean to do that. Okay. When we put it all together, we've got an entire cycle. And we call it a cycle again because when the cell exits, once it goes through cytokinesis and now we've got two new cells, each of those cells will begin their own process of G1. One thing you should notice here is that we've got these red bands in the cell cycle. We've got 3 of them. These red bands represent checkpoints that are extremely, extremely important. If there are no checkpoints, unfortunately, it can lead to cancer. It can lead to unregulated growth of abnormal cells. And if left untreated, and if the body has limited defenses against this unregulated growth, then it can lead to a tumor. The image on the right shows one of those tumors that appears to be a colon cancer tumor. So it's in someone's large intestine. All right. So we need those checkpoints to prevent that cancer from forming. When a cell has some type of error or it is in some way damaged, we need checkpoints to say, okay, you should not enter the process of dividing. So in G1, there's a checkpoint that asks the cell, do you have damaged DNA? Before we make a copy of this DNA, is some of it damaged? Are there mutations? If yes, well, then you should stop right here and you need to go through this process of apoptosis, which is programmed cell death. If everything is good here in G1, then we go on to the S phase. We go into synthesis, where we're making a copy of the DNA. After the copy is made, we need to ask another question. Has some of the DNA failed to replicate, or was some of it damaged during the process of replication? If the answer is yes to that, well, then we can't make a copy of this cell. We need to go ahead and send this cell into apoptosis. If at any point one of these checkpoints is missed, or you end up arriving at the wrong answer to one of these questions, that can be pretty damaging for not just the cell, but for the entire organism. All right, so we need checkpoints to stop growth and division if necessary, and cancer can result if these checkpoints fail. All right. So we've got 18 minutes about left in the class today. There is an asynchronous assignment. There is an exit ticket. You probably have enough time to do both if you're moving and you're working diligently. So I encourage you to go ahead and knock these out. I will be here on mute if you have any questions. Okay, folks, it's now 1:20, so you all are good to go ahead and get ready for fourth block. I appreciate the participation today. I'd like a little bit more tomorrow. We can get back to what we're used to doing, but in the meantime, have a great day, great evening, and I'll talk to you tomorrow.
Have a nice day. See you later.
Peace out. Thanks, Lance.
All right,
Bye-bye.
see you tomorrow.
See you, Jean. See you, Sway. Thanks, JT.